clinically very relevant work, and also for offering me a great transition with the allusion to the clock gene genetics to our next speaker, Charalambos Kyriacou, who is a professor of behavioral genetics at the University of Leicester. He's a highly distinguished geneticist who helped really launch one of the most exciting and important fields in behavioral genetics, which is the genetic control of biological rhythms, and who also very generously shared his personal experience as a person affected by or as a member of a Huntington's disease family at the opening lecture of our conference. So with this, Charalambos, I welcome you to our online session, and I'll hand over the screen to you as we do these days.
Hello, and thank you. Can you hear me? Hello again. I was here a couple of days ago. Some of you might have heard my first talk. I'm a geneticist and I work on circadian rhythms and clock genes, mostly in Drosophila. Now, circadian rhythms infiltrate absolutely every biochemical and physiological behavioral phenotype of every organism that lives on the surface of the planet, and that includes plants, animals, and some bacteria. And Drosophila melanogaster, as I told you a couple of days ago, the genes that control the circadian clock were first identified in flies. And just to run through it very quickly, you have two genes, period and timeless, and two transcription factors, clock and cycle, and they activate period and timeless messenger RNAs early at night in clock cells. These are translated to proteins at the cytoplasm, and then along comes casein kinase 1 epsilon and phosphorylase per, which degrades it. Late at night, period builds up a little bit, binds to timeless, which protects it from the kinase. And timeless is a translocator molecule, and it moves period into the nucleus, where they block the actions of clock and cycle on the transcription of their own genes. So you stop the transcription. At dawn, cryptochrome, which is a blue light photoreceptor, is activated. That physically interacts with timeless. Timeless is degraded, as is per, and that releases the block on period and timeless transcription, and the cycle begins again. And that goes on relentlessly, even in constant darkness, where there's a different method of degrading period and timeless. So that's the clock. You've got two positive regulators, two negative regulators, a photoreceptor, and the modulator. And what this leads to is cycles of per gene messenger RNAs, timeless messenger RNAs, clock messenger RNAs, and their proteins, and they go up and down in these clock cells. And in mammals, the situation is almost the same, except cryptochrome has taken the role of timeless, and melanopsin has taken the role of cryptochrome, and that's the photoreceptor, blue light. Cycle is actually BMAL1, and there's two isoforms of casein kinase that are used. There are 3 per genes transcribed and translated in mammals, and 2 cryogen. But it's effectively the same system with a slight evolutionary tweak. And if you look at the clock molecular topology of not only the main negative feedback loop that we just discussed, but an associated feedback loop which stabilizes it, you can see by the color coding that in flies on the left and in mammals on the right, it's a very homologous system, and that's what won the fly guys the Nobel Prize 3 or 4 years ago. But if you look at the neurons in the fly brain, the pacemaker neurons that generate rhythmic behavior, you find 150 neurons, 75 in each half of the brain, and they're divided into 7 groups, color-coded there. Now, these cycles of up and down RNAs and proteins are in phase in all of these neurons, and they need to be in phase to get a coherent rhythmic output. And the way that's done is through these lateral neurons, these red cells, producing a neuropeptide called PDF, pigment dispersing factor. And this couples all the neurons together, so they're all singing from the same song sheet. Now, if you make a mutation of PDF, what you find is in constant darkness is that the cells are still ticking, but they're ticking in different phases. And what you end up with is a behavioral mess, an arrhythmicity. Now, in mammals, the pacemaker is more complicated. It's the suprachiasmatic nucleus in the anterior hypothalamus, paired structure, 10,000 neurons in each half. And this is the master pacemaker, and it controls all the clocks, peripheral clocks, central clocks, sleep. It controls everything. And there it is. This is the suprachiasmatic nucleus. And what we've got here is a film given to me by a friend and collaborator, Mick Hastings at LMB, of the period 2 gene in the mouse, and it's been labeled with luciferase. And what you see is these beautiful cycles, I hope. It's not working. What you should be seeing is that these cells are rhythmically fading in and out every 24 hours. And I don't know why it's not working. It worked before. And the reason that all these cells are coordinated is because of a polypeptide, a neuropeptide called VIP, vasointestinal polypeptide, which is an analog to the fly PDF. Its receptor, VIP receptor 2, also called VPAC2, is actually homologous to the fly PDF receptor. And if you make mutants of VIP receptor in the mouse, what you find is that here's a wild type mouse in light dark cycles. Its activity is at night. This is wheel running. In constant conditions, it's also got a nice rhythm with activity at night. In the receptor mutant in light dark cycle, the light dark cycle drives the locomotor activity, so you still get nocturnal activity. But in constant conditions, in the absence of the receptor, you see it becomes arrhythmic. The animal becomes arrhythmic. And that's because the cells are ticking, but they're not talking to each other. They're all in different phases. And there are sleep mutants or circadian mutants in humans. I've mentioned this in my earlier talk. So familial advanced sleep phase syndrome, or a morning lark, may have a mutant in the PER2 gene or in casein kinase. These guys are active very early and they go to bed very early. And you have delayed sleep phase syndrome, mutations in period 3 gene, casein kinase 1 epsilon, and in CRY1, there are 2 CRY genes. You can see that these guys are very late. They get up late, they're active, and then they go to sleep very late. But there is another sleep mutation, and that's Huntington's. Here's a normal control, and you can see if this is activity. This is a paper by Jenny Morton some years ago. You can see that a normal patient, control patient, is active and not very active at night, at bedtime. Whereas in 2 patients, Huntington's patients, we have a lot of activity monitored during bedtime, as you can see. And you can also see this in the Huntington's mouse, the R62 mouse. As the mouse gets older, this is the activity of the mouse. This is wild type. This is the mutant. You can see as it gets older, 14 to 15 weeks, the circadian rhythm really becomes very, very ragged, as you can see here in this histogram. So is the molecular clock compromised in the Huntington's mice? Well, here's the suprachiasmatic nucleus, and you're doing in situ hybridization here to the PER2 gene. That's the major clock gene in the mouse. And you can see in wild type in blue, you've got a nice rhythm, and it's slightly blunted in the R62 mouse. Whereas if you look at BMAL1, which is the rhythmic positive regulator, you can see that it's very, very flat in the Huntington's mouse. So clearly the molecular rhythms in the SCN are compromised. Now you can ask, well, is the SCN damaged in HD patients? Well, I've looked at kind of as many studies as I can see in post-mortem brains, and I haven't seen any convincing evidence of any anatomical problems in post-mortem HD brains in the SCN. In other parts of the hypothalamus, yes. But here's a paper by Warmerlin a few years ago, and what they did is they took post-mortem brains and they looked at the numbers of VIP neurons. So it's about, in a control, there's about 1,1700 on average VIP neurons in the SCN out of 10,000 in each half. And what you see is an 85% reduction in the post-mortem HD brains. Now, what does that mean? Well, you saw what VIP does. VIP couples the circadian, the circadian clock neurons in the SCN together. So they all beat in a concerted mode. So if you're going to lose 85% of them, what you're going to have is lots of cells ticking out of phase. And that would lead to the compromised SCN molecular cycling that we see, because the molecular cycling that we see is an average of many, many cells. And if they're all in different phases, that's going to blunt the rhythms. And this could possibly explain the activity that you see in the nighttime of Huntington's patients. And a paper by Chris Colwell showed that the SCN physiological rhythms are also disrupted in HD mice. He used the 97Q model mouse. And you can see, if you look at the spontaneous firing rate, you have a nice rhythm in the spontaneous firing rate in the wild type controls, but not in the Huntington's mouse. And also in the resting membrane, here's a rhythm in the wild type mouse, but that rhythm is more or less gone in the Huntington's mouse. So there's clearly molecular and physiological problems in the SCN. And Colwell's group have implicated a specific potassium channel in this called the BK current. What about fly pacemaker neurons? And here's a Huntington's model fly. What you have here is exon 1 from the human with 120 Qs. And if you look at period and timeless rhythms, you don't see much difference. You see there's a phase difference. You can see the acrophase is significantly different in the mutants. They're delayed. And unfortunately, this study was done in a light-dark cycle. When you see this delay in a light-dark cycle, it would suggest that if you run these in constant conditions, that you'd see a longer period. That's what this would suggest. But unfortunately, these authors didn't do the work in constant conditions, so you don't know. If you look at clock and another clock gene called Vril, another loop, it's a regulator of clock, you see that the amplitude of the rhythms is also blunted significantly. So does this lead to circadian dysfunction behaviorally? But if you look at locomotor activity rhythms, you see morning and evening peaks of activity on every day. Now, what these guys did is they looked at sleep instead of locomotor activity. And the way of thinking about sleep is if you take the inverse of locomotor activity, just inverted that, you get what you call sleep rhythms. And Paul Shaw, 15 years ago, decided he'd do this. He's made a career out of sleep. And it's basically not exactly inverted activity. So you can see in activity, you've got a midday siesta here, and that comes out when you look at sleep too. So when these guys looked at the sleep of these Huntington's flies, what they saw is reduced sleep in the siesta and a lot of reduced sleep at night. And if I invert that, you can see this as a hyperactivity. The flies with 120 Qs were hyperactive. So clearly the fly model and the mouse model are giving you the same story. The pacemaker neurons are molecularly compromised. So flies can be useful in getting interesting information, not just about Huntington's, but also about clocks. I just want to give you one quick example from my own work. And this was done in collaboration with Carla Allerbrandt and Till Ronenberg. They did a questionnaire, they're in Munich, and thousands of people were asked about how long they sleep. And they correlated this in a GWAS study, genome-wide association study, and they found some hits. One hit was a nicotinic acetylcholine receptor gene. On chromosome 12, they had this hit, but they weren't sure whether it was this gene, ABCC9, or KCNJ8, that was the actual target. So I noticed that both these genes are in Drosophila, so I suggested, well, I could knock these out in Drosophila and see if I get a sleep phenotype. And they said, yeah, please do it, because we can't get this published. So here's a control fly. This is day 1, and this is sleep, not activity. So there's your midday siesta, and there's your nighttime sleep, and that's day 1 replotted there. That's day 2, there's your siesta, there's your nighttime sleep. There's day 2 again, that's the controls. And I took 2 independent knockdowns of the ABCC9 gene, and you got a very dramatic effect. You can see immediately that the first half of night sleep is completely missing. In other words, the duration of sleep is 50% of nighttime sleep in these animals. The siesta is not affected. So it looks that in the fly, knockdown of ABCC9 is giving you a sleep duration effect. So it looks as if in the human, the ABCC9 was the target. The hit was in the ABCC9, not the adjacent gene. And this gene actually encodes an ATP-sensitive potassium channel, and that's known to be involved in energy metabolism and in cardiomyopathy. Now, sleep, the inversion of activity, is also being examined by several groups in flies over the years, and a number of mutations have been found. A lot of them are in potassium and sodium channel, Shaker, Sandman, Sleepless, KTP. This is a sodium leak channel. There are ubiquitin ligase and related mutants, Culin-3, another mutant called insomnia. Red eye is a mutation that is in the nicotinic acetylcholine receptor, which supports the GWAS study that I just mentioned. And there are dopamine-related mutants, fumin and tyrosine hydroxylase, which are also implicated in sleep. Now, these mutants have allowed us to examine and identify regions of the brain that are important in sleep. And you'll see here the mushroom bodies, the ellipsoid body, the fan-shaped body, various other neurons. But you'll also notice two clock neurons, the PDF neurons that I talked to you about earlier, and the dorsal neuron 1. So these are implicated in sleep, but it's not just the circadian clock cells that are implicated in sleep, it's all these other structures as well.
Okay, 1 minute left.
Yep, I'm just going to finish now. So can the fly model be useful for circadian studies in an HD context? Well, one thing you could do is work out whether a fly HTT model would alter the expression of the various sleep genes that I've just mentioned. And if so, and some of these genes may be altered upwards or downwards, then you could try and rescue the sleep defects by overexpression or RNA studies. And some of these gene products, especially the channel genes, have agonists, antagonists, and that could suggest a pharmacotherapy. Recently, an Argus mutant, it's a sleep mutant, alters autophagosome expression. And autophagy is rhythmic in flies, and I'm sure in humans, and there's a feedback between sleep and autophagy. So autophagy, which is very important, of course, in Huntington's and is a target these days for Huntington's therapy, could also be important in studying the relationship between sleep, autophagy, and possible effects in the Huntington's model fly. There's also a pragmatic approach in Jenny Morton. If you take sleep-inducing drugs and apply them to Huntington's mice, they'll sleep more. And if you follow them up, they'll have better coordination, better coordination, and better cognitive effects. And finally, there's basic sleep hygiene by HD parents. And that's basically bright lights during the day, darkness at night, no blue light late at night. And that should entrain the clock of HD patients to the environment, and they should sleep better. And that should have beneficial downstream effects. So a simple model, you've got the clock, and that regulates sleep. You've also got the sleep homeostat, the need to sleep. If you don't sleep, you need to sleep more. There's a two-way relationship between these. There's autophagy. The clock regulates autophagy. Autophagy is in a two-way relationship with sleep. Huntington's disrupts the circadian clock with downstream effects on sleep. Huntington's also disrupts autophagy, and that has a reciprocal relationship with Huntington's disease. And of course, a disrupted clock will lead to phenotypic effects in Huntington's as well. So I'll stop there quickly. Thank you.
Thank you very much, Professor Kyriacou, for this really interesting talk.